Now, just like your carbohydrates, just like lipids, proteins are also, your polymers, right? They're also your polymers. So proteins, when we talk about them specifically, proteins are organic compounds consisting of carbon, hydrogen, oxygen. So these are the ones, carbon, hydrogen, and oxygen, that were existing in lipids as well. And they were existing in what? They were also existing in your triglycerides, in your fats as well. Now, in proteins, we have other elements other than these three. Okay. That's one difference that you will see between proteins and carbohydrates and fats. Carbohydrates and fats generally have carbon, hydrogen, and oxygen. Mostly that's it. Proteins, on the other hand, can have carbon, nitrogen, hydrogen, and they can also have nitrogen, and they can have sulfur as well, right? So your proteins are organic compounds that consist of all of these elements. Now, just like carbohydrates and lipids, proteins are also polymer. The proteins are made by joining together the monomer known as amino acids. So the monomer, the building block of proteins are your amino acids. So amino acid is my Lego block. And I can join the amino acids with each other to make my Lego structure, whatever my Lego structure may be. When we talk about proteins, so proteins have a lot of different uses that that have a lot of different uses that make them almost essential in our body, right? What are the different functions of proteins? So the different functions of proteins are that they are, of course, make up our body tissue, right? Most of our body tissue, our muscles, a lot of, your fibrous structure, like your collagen, like your hemoglobin, like the keratin that we have in our nails. All of this is composed of what? All of this is composed of your proteins. So we need them to make up our body tissue. We need them for growth, for development, for repair, and healing as well. We need proteins, right? So they have a lot of different uses. Now, when we are going to inshallah study about hemoglobin and collagen and all in a bit. Now, what we need to understand is how your proteins are going to be made, how the monomers are going to join together. The monomer is known as amino acid. First of all, let's go over the structure of amino acid. What is the structure of an amino acid? So, we will have a central carbon atom. This central carbon atom is going to be attached to a carboxylic acid group on one side. On the other side, it will be attached to an NH2 group. All right. Then on this, the third bond of the central carbon atom is that it will have a hydrogen bond. And the fourth is known as the R group. The R group is basically your variable group. Variable group means that on this side, I can have pretty much anything. I can have another hydrogen as well. That's also possible. Or I can have CH3, or I can have, sulfur present, right? I can have anything at all. I can have hydrogen ion, I can have sulfur ion, I can have nitrogen ion, I can, have any such thing can possibly be present. The R group can be anything, C2H5, anything at all. Okay, so the R group is our variable group. It can be anything at all. Now, why this variable group is very important? Why is the variable group so important? Because it gives certain properties to the amino acid. Okay, it gives a certain property to your amino acids. Now, those properties are important because when I talk about the structure of your protein, if you're going to take a look, you're going to see that different proteins have got different functions. Now, question that you can ask me is how does that happen, miss? How is it that one protein is responsible for carrying your transporting oxygen while the other protein is providing strength and support? Why are we seeing this difference? The reason we're seeing this difference is because we can have different types of amino acids. There are around 20 different amino acids. Okay, we have around 20 different amino acids. So, and what is the difference between those amino acids? The difference between those amino acids is only the R group. The rest of the structure is the same. So only the main difference that we have between the 20 different amino acids is the R group. The R group is different. So because the R group is different, the amino acid will give different properties to the protein. How does it do that? How does the different R group actually lead to different functions of the protein? That we will understand in just a bit when we go over the structure of proteins in a bit of detail. But, so this is what the structure of an amino acid looks like. Okay, fine. Now, how do the amino acids join up? Because so far in carbohydrates and also in fats, we talked about how the bonds are made, right? So how are the bonds going to be made between two between two amino acids? Let's take a look. This is my amino acid. On one side, it will have NH2 bond. On the other side, it will have a carboxylic acid bond. Right? Then on one side, it will have hydrogen. And on the other side, it will have your R. This is one amino acid, and then we have another amino acid. Now, what's going to happen? A bond is going to form between the hydrogen of your this. Hold on one second. Between the hydrogen of your amine group and the OH of the carboxylic. A bond is going to form between nitrogen and carbon. The bond between nitrogen and hydrogen will break, and the bond between carbon and OH group will break. The final result that you're going to end up getting, your structure is going to end up looking something like this. You're going to have, let's draw the first amino acid. This is my first amino acid. Now, this first amino acid has an R group, hydrogen. I will draw the COH on the side over here. Now, the bond is going to form between nitrogen and the carbon of the second amino acid. Over here, I will have another oxygen. Then the rest of the structure of the amino acid will be drawn. This bond that you are seeing over here, this bond is going to be known as your, this is the bond that forms between amino acids. And this bond that forms between amino acids, this bond, we call it peptide bond. So just like with triglycerides, it's ester bond. Just like with your carbohydrates, it's glycosidic bond. So this is going to be known as a peptide bond. The bond that will happen between the two amino acids is going to be known as a peptide bond. I will also get a molecule of water in the end, right? Because the hydrogen is reacting with the OH. I will also get a molecule of water in the end. This reaction is known as my condensation reaction. I can also do the opposite of this. What is the opposite of this? The opposite of this is that I have my protein and I add water to my protein. So I can break down the peptide bond as well. The reverse reaction is known as hydrolysis. So I can reverse the reaction by addition of water. I can break down the peptide bond and I can go ahead and I can make my two amino acids. Okay. So this is my amino acid number 1. And this is my amino acid 2. And the two, so over here, amino acid, 2 of the amino acids have joined together. When two amino acids join together, the structure that is formed is known as a dipeptide. Why do we call it a dipeptide? We call it a dipeptide because there are two amino acids present. Now, if I continue to add amino acids to it, if I continue to add more and more amino acid to it, then it will become your polypeptide. If I continue to add more and more amino acid to it, we will call it a polypeptide or we will call it a protein. Okay. So when two amino acids join with each other, it's known as a dipeptide. And when more than two amino acids join together with each other, we are going to be calling it a polypeptide. So this is how your amino acids are actually going to join with each other to go ahead and actually make your bond that you see in your protein structure. Making sense so far? Anything that you would like to ask?
No, everything is clear, but can I
Yeah.
ask just one thing?
Yeah, go on, go on.
The R group, what is it made of? Like, is it the sulfur and the nitrogen atoms?
It's going to depend on the it's going to depend upon the type of amino acids. Okay. In some amino acids, the R group can be made out of sulfur. In some amino acids, the R group can have CH3. In some amino acids, you might have just another hydrogen. Okay. So
Mhm.
each amino acid will have something different. It
Okay.
can be, yeah. So it can be anything. It can, but it's going to be something different. So each amino acid is going to have something different on it, and that's the reason. Because each amino acid has something different on it. So just to like go over an interesting concept with you, just because you know each amino acid has a different R group because it has something new in the R group, something different than the other ones do not have. So that gives it specific properties. And that is why whenever you have like a mutation in the DNA, so what happens that if the DNA will change, then the amino acid that we will, the amino acid that will be used to make the protein will be a different amino acid. And we're going to do this topic in anyways in I think the next unit. So the amino acid we will use is going to be different, right? Because the genetic information has changed. So, what actually happens as a result of all of that is that now, why is the wrong amino acid being chosen so bad? The reason having that your wrong amino acid being chosen is so bad is because if the wrong amino acid is chosen, then what is going to happen? The R group will change. The R group will change, so the properties of amino acid will change. That will change the properties of the protein, and the protein will not be able to function anymore. So that is actually the main thing over here, that all the R group are going to be all different. Someone is going to have the sulfur group in their R group. Someone will have maybe just one methyl group, CH3. Maybe someone will be C2H5. Maybe someone will only have hydrogen, right? So different type of amino acid will have different type of R group. And that is what makes the situation a bit complicated, because different type of R group means different properties. So if we incorporate, or if we include the wrong amino acid in the protein structure, the properties of the protein will change. And right now, just in a little bit, actually just in a little while, it will make more sense that how can the change, or how can the R group have such a strong effect? How does the R group actually have such a huge effect on the properties of proteins? That will become clear in just a bit. But does that answer your question? Making sense?
No, yes, it does.
All right, great. Now, when you're going to go ahead and you are going to talk about it, when we talk about our structure of protein, okay? So the structure of protein can sort of have four levels. So it's not, I'm not saying that it will be like four levels, like level one, level two, but sort of like think about it, degree of complexity, okay? So we can have the least complexity and we can have like the most complexity. Think of it that way. That's not how you will explain in the exam, but just for you and me to understand, we're going to talk about complexity. Okay. Now, when I start to make the protein, the first step is to join the amino acids with each other. That is known as the primary structure. Primary structure, what is the primary structure of protein? It is simply a chain of amino acids joined together by the joined together by what? Joined together by the peptide bond through the condensation reaction. That's it. So if I just take my amino acids and I join them together to give me a straight chain structure, right? To give me a straight chain structure, that is going to be known as my primary structure. That's it. So in the primary structure, what type of bond is seen? Only the peptide bond is seen, because I'm going to teach you other bonds as well in a bit. So we need to know in which structure, what kind of bonds we will see. So a primary structure is simply when I will take my amino acids, I will join them each other in a particular sequence, in a specific sequence. The sequence is important, because the sequence, as you will see in a bit, the sequence will affect the structure of the protein and the function of the protein. And who will determine the sequence? The DNA. The DNA will tell me what is the correct amino acid and what is the sequence as well. It will tell me both things. So if I take the amino acids, put them in a particular sequence, make a straight chain out of it, and join them together by the peptide bond, that is known as your primary structure. The primary structure of the protein, if I draw this out for you, it looks like, quite simply, it looks like this. It looks like this. This is, for example, my mm, for example, one amino acid is known as cysteine. We can have cysteine is the amino acid that has sulfur in it. Okay. So the amino acid that contains sulfur is known as cysteine, by the way. So I have cysteine. Cysteine has made a peptide bond with the next amino acid, that is known as glycine. The variable group of glycine is simply a hydrogen atom. That's If the R group has one hydrogen atom, then that amino acid is known as glycine. Glycine is the smallest amino acid that we have. Then, if the R group has sulfur in it, that is known as cysteine. Okay. Then the rest of the amino acids can have different things. Okay. So we can have, for example, cysteine. Then there is another amino acid that is known as lysine. Another one that is known as, for example, proline. We can have leucine, we can have isoleucine. So there are over around 20 amino acids. If you start, you don't need to learn their names. But the point is that if I take these amino acids and I arrange them in the specific sequence as dictated by the DNA, and I join them by a peptide bond to give me a straight chain of amino acids, that will be known as the primary structure. Now, once my primary structure is ready, I will call it a peptide chain, or I will call it a polypeptide. I will not call it a protein yet. Why? Because a protein generally can have further processing. Generally, not necessary, but most of the time, to form a fully functioning protein, I need to do further processing. What do I mean by processing? Folding. Okay. So I can take this straight chain and I can fold this straight chain to give me a secondary structure. Secondary structure is I've taken my primary structure, I've taken the chain of amino acids, and I have made it undergo further folding. I have now started to fold up the amino acid. That's a protein, sorry. That's why I said it's about complexity. This is the least complex straight chain. Then I can start to fold it up and make it a bit more complex, right? How can I make it a bit more complex? So in the secondary structure, hold on. In the secondary structure, what is going to happen, that I can further fold this chain into possible forms. Okay. One possible form that we have is known as alpha helix structure, and the second possible form that we can have is known as beta pleated sheets. Let me explain this to you. Okay. Can be of two types. We can have A, alpha helix, or we can have B, which is beta pleated sheets. Now, what happens that we have we can develop hydrogen bonding. Okay. If you're doing chemistry this year, you're going to study hydrogen bonding in a bit of detail, like exactly how it is made. But just to go over the general, like very basic concept with you, what is hydrogen bonding? Hydrogen bonding is a bond. There are two conditions to it. Okay. The two conditions to it are that one, I need a hydrogen, and that hydrogen needs to have a covalent bond with either oxygen or nitrogen or fluorine. The second requirement for hydrogen bonding is that I need what? I need another molecule in that same area. So I need one molecule that has hydrogen on it, and then I need another molecule that has oxygen or nitrogen or fluorine that has a lone pair of electrons. So what happens in the amino acid? What happens in the amino acid is that this is my carboxylic acid. right? This is the rest of the amino acid. Now, what is going to happen? I have this carboxylic acid. Now, in this carboxylic acid, um, what is going to happen? See, I have this hydrogen that has a bond with oxygen. Now, what is going to happen that when you do chemistry, you'll come over a concept of electronegativity. What is electronegativity? Electronegativity is the ability of any atom to attract the electrons towards itself. Let me be a bit more clear. To attract the shared pair of electrons towards itself. So, for example, we have two atoms that have a covalent bond. One of the atoms that has more ability, that has more electronegativity, it can pull that shared pair of electrons towards itself. That doesn't mean that it will take them completely. No, it's not an ionic bond. They will still be sharing it, but it's just that, they will sort of shift, they will sort of move towards one of the atoms. So they won't, it's not like an ionic bond. It's not like it will take those electrons. It's just that, it will sort of pull them towards itself because it sort of, has a bit more ability. It has a bit more attraction towards the electrons. So that is what oxygen will do over here. Okay. So the oxygen ends up developing a small, teeny, tiny little charge. That small, teeny tiny little charge, the term used for it is partial charge. Partial charge means that it's not like a permanent charge, not like an ionic bond. This is just, due to sharing of electrons, just, shifting of electrons. One is becoming slightly negative, partially negative. So oxygen will be partially negative. Hydrogen will have a bit of a positive charge. This is one molecule of amino acid. Then let's say I have another molecule of amino acid. Okay. Now, what is going to happen that between the what's going to happen that over here I have my partial positive hydrogen. So this is the hydrogen that has slightly positive charge because all the electrons have gone to oxygen. Then this is the oxygen that is slightly negative because it has attracted all the electrons from the carbon towards itself. So a bond will form between this hydrogen and between this oxygen. This is your hydrogen. So hydrogen bond is going to form. So in our secondary structure, we will see two types of bonding. Okay? We will see two types of bonding. What are the two types of bonding that we're going to see? It has two types of bonding. One is, of course, the peptide bonding. That won't go anywhere, right? Peptide bond is going to stay exactly where it is. It's not going to go anywhere. It will be in the same place because it's holding all the amino acid together. If I break the peptide bond, the amino acids will go away. Keep in mind that the peptide bond is a strong bond. It's the covalent bond. This is just a hydrogen bond. Hydrogen bond is one of the intermolecular forces of attraction, weak intermolecular force of attraction. It's not that strong. Peptide bond is strong. So one is peptide bond that's holding the amino acid together. And the second one is hydrogen bond that will form between the partial positive hydrogen in OH group and the partial negative oxygen that has your C double bond O, right? So what is going to happen? That they're going to end up making hydrogen bonds because these hydrogen bonds are going to start forming between. So glycine, for example, will make a hydrogen bond with proline. Proline will make a hydrogen bond with leucine, like that. So the amino acids, first of all, they're arranged, joined to each other with the help of what? They're joined to each other because with the help of your peptide bond. Then on top of that, they start developing hydrogen bonds with each other, right? As a result, the molecule will start to fold. Now, the folding can happen in two possible manners. One possible manner that you can have is you can have sort of a spiral structure. What is the spiral structure that I'm talking about? You can have, for example, something that looks like this. So the molecules, this is, for example, your amino acids. This is the chain. You can get a spiral structure like this. And these are the amino acids, let's say. These dots are your amino acids. So these dots can develop hydrogen bonding with each other. These dots can develop, these amino acids can develop hydrogen bonding with each other. And because of this hydrogen bonding, they can sort of end up having a folded structure, a spiral structure. This spiral structure is what we actually, when you're going to read your, when you're going to look into it, the name for this that we give is that this spiral structure is what we call as our alpha helix. Similarly, these amino acids can also develop the bond in such a way that they can develop sort of a pleated structure like this. They can develop a pleated structure like this. So it will literally look like, it has pleats like this, right? This kind of a structure. So this is, for example, these are my amino acids. So you will end up getting hydrogen bonding between the amino acids. Again, just like I explained to you, in the same way, I will get the bonding. Now, the type of bonding that is going to be taking place is, going to depend upon the structure of the protein and all of those things. So what's the function of the protein? What's the structure of the protein? So which structure ends up forming is going to depend upon the protein. But this sort of a structure would be known as beta pleated sheets. So your amino acid can undergo folding in two different ways. Either it can make a spiral or it can make what we call a beta pleated sheet. Making sense? Any confusion?
No, no, it's okay so far. Just
So
a
far,
bit
it's fine.
too much, yeah.
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 All right. Okay, fine. So if this is all clear, then I can take this structure and I can make it even more complex. Meaning that I can take this structure and I can make it even more folded. I can introduce different types of foldings to it, right? Now, that will make the protein a bit more complex. Okay. Now, by the way, just to clear one thing up, the type of structure your protein has is going to depend upon the requirements of that protein. Like, what's the genetic information saying? What is the requirement for that particular protein? It's based on that. So, some proteins, for example, the maximum level of complexity they can have is secondary structure. And we will study those proteins, by the way, as well. So, there's certain proteins for them the maximum level of complexity that they will show you is that they will give you is that they're going to go ahead and they're going to give you your sorry they're going to go ahead and they're going to give you what they're going to give you just the secondary structure, that's it. They won't give you the next level. So I just want to make that clear that it's not necessary that you are going to see all these levels in all proteins. Some proteins might only go up to the level of secondary structure. Some of them might go all the way up to the last complexity. So it's not necessary that all of them have the same amount of complexity. They can have different amount of complexities as well. Okay. Now, after this, I can have the third level of folding, which we call as tertiary structure. Tertiary structure is what is sort of, when we're going to talk about it. So it is okay. Our secondary structure, by the way, is also 3D structure. Okay. So our secondary structure is also known as 3D structure. So our tertiary structure is one step ahead. Like I've taken the 3D structure and I have further modified. Okay. So this is our 3D structure that has undergone more folding to make the protein compact and spherical and globular. Okay, so I can take that. So I can take that protein, I can take my secondary structure, and I can make it undergo even more foldings. I can fold it up, I can wrap it around in such a way that it sort of becomes, a bit spherical. It becomes like a globe, it becomes like a compact, small, teeny tiny protein. And we will study what kind of proteins have this structure in just a bit. So we can have something like that as well. Now, when we're going to go ahead and talk about it, now how you can ask me, okay, now what? You have already joined the amino acids together, and then you have also made them develop hydrogen bonds. How can we make the structure even more complicated? How can I, make it like even more, right? How can I fold it even more? What else can happen now? So there are multiple bonds seen in tertiary structure. What are the different bonds that we will see in tertiary structure? One, of course, when we're going to go ahead and talk about it, one of them is your peptide bond, right? Same as before, and the hydrogen bond. These two are carrying forward. Other than these two, we can now have different types of bonds. One is known as sulfide bond, or we use the term sulfide bridges. Then we can have another type of bond that is known as ionic bond. Then we can have another type of bond that is known as our hydrophobic interaction, right? So now we have already studied about hydrogen bonds and peptide bonds, so I will not go over them again. Do let me know if you want me to revise, but since we've covered them before, I won't be going over them again. So we are going to take a look at we're going to go ahead and take a look at our if, we are going to take a look at, for example, let's talk about disulfide bonds or disulfide bridges. Remember, I told you the amino acid that contains sulfur is known as cysteine. Okay. So this disulfide bond will only form between the amino acids that have sulfur. So that is cysteine. Cysteine is the only amino acid. Forms between the sulfur containing R group of cysteine. Cysteine is that amino acid. The R group of cysteine has sulfur in it. So it forms between the R group of your cysteine. Okay, the R group of cysteine has sulfur in it. So it forms between that, right? So what is the reaction that occurs? The reaction that occurs is basically it's an oxidation reaction. It's an oxidation reaction, right? Resulting in a strong covalent bond that is known as what? That is known as our disulfide bridges. So these disulfide bridges are going to be really important because they're going to help in keeping our molecule in further shape. For example, I have my, okay, let me draw the protein structure first. Let's say this is my protein structure. Okay, see. So this is my protein structure. In some areas it has gone, a bit of alpha helical shape. Then in some areas it has undergone beta pleated sheets. Then again in certain areas it has got hydrogen bonding, something like this, right? So what can happen that we can now also end up seeing that disulfide bridges or disulfide bonds can form as well. So let's say I have an amino acid cysteine, it has to be cysteine over here, and I have an amino acid cysteine over here. So the strong covalent disulfide bridge can form over here as well between these two amino acids of cysteine, right? So see how that is helping in the folding of the protein. And this is also cysteine. So we can end up getting our disulfide bridge. So disulfide bridge is a strong covalent bond, which means it normally doesn't break very easy. You need a lot of temperature change. You need a lot of your temperature change to take place. To if I want to break this, I will need temperature change and stuff like that. When we talk about hydrogen bonds, keep this in mind, hydrogen bonds usually break easy. Okay? Meaning that if there is going to be a change in pH, if there's going to be a change in temperature, hydrogen bonds are going to break pretty easy. They're not very strong. So this is our something what this tertiary structure is going to look like, right? So now we have developed disulfide bridges. Then, so they will form between cysteine. And what is cysteine? Cysteine is an amino acid that has an R group which has sulfur in it. Okay, and the bond looks something like this. If I draw the chemical formula for you, so the bond looks something like this. This is sulfur, and then this is sulfur, and then of course you will have your carbon and your carbon, right? All right. Then if we continue talking about it, so the next bond that I mentioned to you was we can also have ionic bonds. Ionic bonds will also form between R groups. Okay, keep this in mind. So now this disulfide bridge, ionic bond and hydropic interactions, they form between R groups. Okay, peptide bond happens between the amino acids, between the carboxylic group and the amine group. Hydrogen bond is also with the carboxylic acid between the oxygen and the hydrogen. But disulfide, ionic, and hydropic have nothing to do with carboxylic acid group and the amine group. They have everything to do with only the R group. This is why the R group is important. If, for example, over here we had the amino acid cysteine, I accidentally, or because of mutation or because of change in DNA, I put some other amino acid like glycine, which doesn't have sulfur in it. So it will not be able to make the disulfide bond because it does not have the correct R group. The protein will not fold properly, it will not work properly. This is why I was saying that the R group is so important. Then we can have our ionic bonds. They can form between positively charged and alhamdulillah negatively charged amino acids. Sorry, I will be very specific. R groups of amino acids. So we have certain amino acids that have, when you're going to go and take a look at the structure, you're going to see that those amino acids, that the R group has a negative charge or the R group has a positive charge. So the R group having a negative charge, that R group having a positive charge is really important because the positive and the negative charged R groups will be able to develop what? They will be able to develop a bond with each other. Okay. So for example, over here, if we take a look, this is just an example that it's not necessary that this is how it is, but this is my amino acid. This is my chain. Okay. Now my chain continues the folding. And for example, it goes ahead and it develops we have a over here and we have over here. Okay. This one, for example, has H plus. And this one, for example, hold on, give me a second. Right? We have an H plus over here. And then on the other side, let's say we have some O negative ion. Okay. So a bond can form between the H plus and the negative ion. This would be known as your ionic bond. So ionic bonding is again really important. And ionic bonding is also generally broken down easily when it comes to your heat, right? Heat, so hydrogen bonding is affected both by pH as well as your temperature change. Similarly, ionic bonding are also affected quite easily with the help of heat energy, right? Then, for disulfide bridges, generally we need to use some kind of chemicals. Okay, this is why your enzymes get denatured when we change the temperature and pH, because enzymes are made up of proteins. By changing the pH and by changing the temperature, I will break the peptide bond, I will break the ionic bond, and as a result, what is going to happen? The structure of the enzyme changes, so it is not able to function anymore. So then we have, and then we can have hydrophobic interactions. What are hydrophobic interactions? So we have, if you're going to take a look, there are certain R groups contain hydrophobic groups. Hydrophobic groups are water hating groups, right? So, for example, a lot of your alkane type of group, CH2, CH2, CH3, sort of this type of a group, which is a lot of alkanes in it, a lot of alkanes in it, no oxygen, nothing like that. Just, an alkane, CH2, CH2, something like that. So that would be hydrophobic, right? So those R groups are hydrophobic. So these hydrophobic groups can develop weak bonding with each other. How do they develop weak bonding with each other? They develop weak bonding with each other. If you remember when I told you about lipids, I told you that the hydrophobic tails would all point towards each other, right? So they can develop weak bonding with each other so that they are all facing the inside of the molecule or the inside of the protein. So they will try to arrange themselves in such a way, they will try to develop interactions in such a way that they are all facing, all the R groups are facing the inside of the protein, so they can be away from the water. That is the reason why the tertiary structure can have, a spherical shape, a ball-like shape, because all the water loving amino acids, they can be on the outside, all the water hating amino acids can sort of come towards the inside of the protein, right? So for example, my hydrophobic amino acids, when I'm going to go ahead and talk about it, they will all, for example, I have a hydrophobic amino acid over here and over here, so they can develop interactions with each other in such a way that they are towards the inside of the protein and not towards the outside, right? So these are all the different type of interactions or bondings that you're going to see. The overall result of this entire story is that at the very end, I have my tertiary structure. And if you will take a look, you will see it is so much more folded compared to the secondary structure. Compared to the secondary structure, it has undergone a lot of folding, right? And the reason is because of all these different bonds that have taken place. And why do we have all of these different bonds? Because of the different R groups. If the R groups were incorrect, then the situation would be completely different. And last, but not least, level of complexity that we can see in our protein structure is what we call our quaternary structure. Quaternary structure is very easy. After all this, quaternary structure is very easy. Now you will ask me, Miss, what more can happen? I've taken that protein and I've folded it every single way possible. Quaternary structure is when more than one peptide chain join. That's it. So I have got two tertiary structure, two protein chains that are in tertiary structure. Okay? Two tertiary chains that are in tertiary structure. Okay? So what is going to happen? Those two chains both have tertiary structure. They ended up, developing hydrogen bonding, ionic bonding. All the bondings I taught you, they are the same. Quaternary structure has the same bondings as tertiary structure. So because of hydrogen bonding, because of ionic bonding, because of whatever bonding, those two chains join together. They didn't join by peptide bond. Keep that in mind. Hydrogen bond, ionic bond, disulfide bond, something like that. Those two different chains of protein join together. Till tertiary structure, we were talking about one chain. It was one chain being folded and folded and folded and folded. Now we have two chains, or maybe three chains, or maybe four chains, all joining together to make one full protein. That is known as quaternary structure. And this is what we see in hemoglobin. Hemoglobin is a quaternary structure. It's made up of four proteins, right? So for example, this is one of my tertiary structure, right? It's undergone all this kind of folding. This is one complete chain, okay? Then I get another chain. Similarly, that is also undergone various, alpha spirals, whatever the structure may be, right? Whatever the structure may be. Now, these two, because of your hydrogen bonding, ionic bonding, whatever type of bonding, these two have joined together with each other. Keep this in mind that they won't develop peptide bonds, okay? Some hydrogen bonding, ionic bonding, hydrophobic interactions, whatever, doesn't matter. But because of these interactions, these two have gone ahead and joined together with each other to make your final protein. This final protein is what we're going to go ahead and consider as our quaternary structure. So quaternary structure is simply when I will take two of the tertiary structure or two of my polypeptide chain, and they will develop interactions, certain interactions that will go ahead and that will allow it to actually that will go ahead and allow it to make one complete protein. That is what we're going to consider as finally our quaternary structure. So the degree of folding or the degree of complexity is going to again depend upon what kind of protein am I making. In hemoglobin, I will end up seeing something all the way to my I will end up seeing all the way up to my quaternary structure. I will see all of these complexity. I will see simple chain. From simple chain, it will fold all the way until it gets to the stage where it is a quaternary structure. Okay. On the other hand, my enzymes will only get up to the stage of tertiary structure. Most enzymes only get up to the stage of tertiary structure. They don't even get, they're not, there's just one chain that's folded together. Then we have certain proteins that will only be a maximum level is secondary structure, like your collagen, like your keratin, a lot of, your structure proteins that provide us strength and support. They generally only, they will only fold up to the level of secondary. They won't go past secondary. So we can have different levels of or different types of bonding that is seen in our protein. So proteins can develop different level of structures. All clear? Any confusion? Anything that you would like me to go over?
No, everything's clear, but
Mm-hmm.
let's go over the
Yeah, yeah.
hydrophobic interactions.
Don't worry about the hydrophobic interactions because we are going to, in the next class, we're going to do the topic of globular proteins. So I will explain to you over there because
Oh,
we have to do this in detail when we do that anyway. So then we will do this in detail. We will discuss
mm.
about what hydrophobic interactions are. So
Okay.
yeah, this was just me giving you like the name and what they act like, just an overview of it. We have to study them in detail anyway. So when we do next class, I will go over in detail overview with the hydrophobic interactions. Anything else that you need to ask? Is everything else all clear?
No, everything is clear.
Okay, great. Okay, then, Asma, we're going to go ahead and stop it over here for today. And inshallah, we will finish off with the topic of proteins in the next class. All right?